Hello friends, myself Dr. K. W. Pawar from Department of Botany, रैत शिक्षण संस्थाज R.B. Rao Borake, College Srirampur, District Ahmednagar. Today we are going to see the practical, or today we are going to perform the practical from first year B.Sc. There is the paper that is Morphology and Anatomy, BO 112. The practical is based on the Morphology and Anatomy, BO 112, paper second. So, what is the aim? Aim is of practical, that is study of internal structure of monocotyledon stem, or what we call the monocot stem. For instance, there is the example of the meristem or what we call the geomes. Okay. So, today we perform the section cutting for the internal structure. Section cutting, then observation under the slide, and then the particular what are the characters that are seen in the particular microscope. So, today we observe in the particular section. So, first of all, we are going to take the section of the particular stem. Okay. So, here we are going to study the internal structure of the particular meristem. Friends, these all pieces is of the particular meristem. You can see here in petri dish. These are the pieces made of the particular meristem. So, I will choose one of the piece of young stem and I will cut it on the particular transverse section. Okay. So, first of all, I take the particular meristem and here I cut the particular meristem in the transverse section, or what we call it as the TS. I take the 4-5 transverse section of the particular thin sizes, because if it is thin, then we can identify the all parts. Here you can see there are the 5-6 sections are there. I take the 5-6 sections are there. Here the precaution is taken that you should use the newer blade, that's so that there will be the section is again the very thin. I choose the one of the section from this 5-6 section of the thin size. Okay, I choose this section of the thin size. Then I remove the water and then it stained by the, this is the stain that is of the saponin. I will stain this particular by putting the one drop of the saponin in the on the slide, and then there is the particular I will take the another slide for the particular section. Before that, the section will wash in the water so that to remove the excess stain from the particular section. Then when you handle the section, that time you compulsory require the brush, because the section is very delicate one. So, there is the requirement of the particular brush. So, again I remove the particular water from the section, and there is a 1 or 2 drop of the particular glycerin, that is the mounting medium, I place on the particular section. And then after that, with the help of cover glass or what we called cover slips, I will put the cover slip on the particular section with the help of the needle. Here just like this, I will put the cover slip so that you can see here center in the section of the slide. Then after that, these sections, I will observe the section on the particular microscope. It required the compound microscope. First of all, I am going to focus the microscope and focus this particular compound microscope, and then I am placing the slide on the microscope, and there will be the observation of the particular material. Okay. So, one can observe the material under this particular microscope. I focus the slide under the microscope, and whatever the part we will see on the particular chart. We will now see the part, whatever the part that are seen under the microscope, we will see on the particular chart. Okay. Okay. After placing the section under the microscope, one can see these particular like this structure. Friends, this one is the particular ground plane of the section. Just I take the transfer section, and this one is the ground plane of the particular section, and this particular view is of the sector view of the section, like this. Here when we cut the section in the particular sector view, the particular sector view see like this. So, on the chart there are the three types of the figure is here. This one, first of all, there is the ground plane, then there is the sector view of the section, and this one is the vascular bundle, individual or single vascular bundle, magnified single vascular bundle you can see here on the particular chart. So, the sector view consists of the outermost layer, that is the epidermis, then there is the particular next to epidermis or below the epidermis, there is the particular hypodermis, or what we call epidermis, then cortical region, cortex. Cortex is made up from the hypodermis, and there is the particular ground tissue. Here in center, all the particular center that are accumulated by the particular ground tissue. So, and in the ground tissue there is the presence of the vascular bundle or vascular bundle that are scattered in the particular ground tissue. So, the stele is called as the particular atactostele. Stele is called as the atactostele because the in case of the particular dicot, the vascular bundle that are arranged in the ring. Okay. Vascular bundle that are arranged in ring, but in here monocot there is the vascular bundle that are scattered in the particular ground tissue. When we going to see the particular sector view of the stem internally, we can see here there is the epidermis, then cortex, and there is the ground tissue. Then epidermis is epidermis, then hypodermis. Cortex consists of the hypodermis and the particular, these all are the ground tissue. Here 2 to 3 layered hypodermis is there. So, epidermis is single layer. There is the compactly arranged cell. You can see here in the figure, there is the cells are compactly arranged, and above the epidermis there is the particular, here you can see there is the cuticle or thick walled cuticle is there, and sometime the stomata is seen on the, this one is the small minute pore, what we called it as the stomata. Stomata is seen on the particular epidermis. What is the function of stomata? Function of stomata is the exchange of the gases, or it for work in the particular transpiration purpose. Okay. Then what is the function of the epidermis? Epidermal layer protect the internal organs of the particular particular plant body, or it protect the insect and pest attack to the internal organ of the plant body. It also protects the mechanical injuries to the internal organs of the plant body. Then just below the epidermis, there is the hypodermis. 2 to 3 layered hypodermis layer is there. Again, there is the cells are the compactly arranged. The cells of hypodermis is made up from the sclerenchymatous cell. The cells you can see here, there are the 2 to 3 layer sclerenchymatous cell. Like in dicot, there is the endodermis, pericycle, vascular bundle. These are the absent because due to the scattered vascular bundle in case of the monocot. Then 2 to 3 layered hypodermis is there, and in all the particular region there is the particular ground tissue. The ground tissue is again the particular parenchymatous one. They are loosely arranged. You can see here there is the intercellular space between the cell. So, the cells are the parenchymatous one, loosely arranged, and what we call it as the ground tissue. Then about the vascular bundle. In case of monocot stem, the vascular bundle is conjoined, collateral and closed type. This thing always keep in mind, conjoined, collateral and closed type, because why we call the closed type of the vascular bundle? Because there is the absence of the particular cambium ring in between the vascular bundle or cambium strip in between the xylem and phloem in vascular bundle. So, what we call it as the closed type of vascular bundle. Suppose it is present, cambium strip is present in between the xylem and phloem, one can call the open type of the vascular bundle. But here this is the totally absent. So, vascular bundle are conjoined, collateral and closed type. They are scattered over the particular ground tissue. Then in the central vascular bundle is the bigger one as compared to the other vascular bundle. At the center there is vascular bundle, and that particular vascular bundle is the bigger as compared to the other vascular bundle. Then here there is the vascular bundle, magnified image of the vascular bundle. Vascular bundle made up from the particular xylem and the phloem. Vascular bundle made up from the xylem and phloem. Here when we going to see the xylem and phloem, there are the two types of xylem element are found. One is the bigger element of xylem, what we call the metaxylem, and smaller element of the xylem, what we call it as the protoxylem. Okay. You can see here there is the big element of the xylem, and that's why the xylem is the Y-shaped. Okay. When we draw the such type of the line, then you can see there is the Y-shaped xylem is there here present in the monocot stem. Okay. The xylem is made up from the vessels, tracheids. Vessels, tracheids, you can see here there is the pitted vessel, then annular vessel, and there is the spiral vessel. So here there is the particular xylem parenchyma. You can see here there is the presence of also the xylem parenchyma. You can see here there is the water containing cavity. Protoxylem regenerated. Sometime there is the protoxylem get regenerated and it forms the cavity. It forms the cavity. That cavity what we called water containing cavity, or what we called it as the lysogenous cavity. It is called as the lysogenous cavity, or sometime it is called as the water containing. This cavity is formed by the regeneration of the particular protoxylem element. Then there is the phloem. Phloem is made up from the sieve tubes and the companion cell. One thing you keep in mind that phloem is made up from companion cell and sieve tube. The phloem parenchyma is altogether absent in the vascular bundle of the monocot stem. Okay. You can see here this one is the protophloem broken, then these are the companion cell and these are the sieve tubes. So, the phloem is on the particular upper side or xylem toward the particular center and phloem toward the particular periphery, or what we call the toward the epidermis. This type of arrangement here in the monocot stem. Then this one is the bundle sheath. This one is the bundle sheath, or one can call it as the covering of the particular vascular bundle. Bundle sheath, or what we call the covering of the vascular bundle, that particular bundle sheath made up from the sclerenchymatous cell. Bundle sheath that are made up from the sclerenchymatous cell. So, this is about the vascular bundle, and the monocot stem shows this sort of the particular plane or structure: epidermis, hypodermis, then there is the particular ground tissue. Vascular bundle consists of the xylem and the phloem. So, this is the overall scenario of the internal structure of monocot stem, for example, giamase. Okay.